If we talk about the Rubisco enzyme just a little bit more, the Rubisco enzyme we said is going to be the first enzyme that really fixes the carbon when we're talking about photosynthesis. However, that Rubisco enzyme did have two different types of activities. It had the carboxylase activity, and that was what the C stood for. And carboxylase is what happens when it binds CO2, and that leads to then the Calvin cycle being run. And that's a beneficial thing for a plant because that's going to then produce some sugars that can be utilized. The other function that Rubisco has is the oxygenase function. The oxygenase function is a different function, but this is what occurs when we have a different molecule that binds in the active site. So this is what's going to happen when Rubisco binds oxygen gas. If the Rubisco binds oxygen gas, we do not get the Calvin cycle, but instead we get a different process which is called photorespiration. So we do actually have two different molecules, carbon dioxide and oxygen gas, that are really competing with each other for the active site of Rubisco. And depending on which molecule binds in the active site, we're going to have a different activity or process that takes place. If we look at the photorespiration, it is not photosynthesis and it's not respiration. So we don't want to get it confused with those two specific processes. This is going to occur when the Rubisco binds the oxygen gas. And when this occurs, it is going to break down the sugars that are inside the plant. And as it breaks those sugars down, it will be consuming ATP to make that happen. So that's somewhat where the respiration part of the name comes from, that it does break down sugars. However, we're using ATP to do it and we're not generating any energy. So it really is a negative thing to have taking place inside a plant. It is undoing what was just accomplished by photosynthesis. So it is reducing the overall rate of photosynthesis for sure. Now, since we have two different molecules competing for the active site, whenever we have a competitive inhibitor, it can be outcompeted. So it can be outcompeted if we increase the amount of the substrate that we want to bind. So if we want Rubisco to bind carbon dioxide, which would certainly be what a plant would want to happen, then we need to have a higher concentration of carbon dioxide. Now, if we go back to the metabolism that we talked about already, the plants that just run through the light reactions in the Calvin cycle as we described, those are what we call C3 plants. They're called C3 plants because the first stable organic molecule that we get is a 3-carbon molecule. There are other alternatives. So C4 plants are plants that have special adaptations that are going to enable them to increase that concentration of carbon dioxide. Because they increase the concentration of carbon dioxide, we will have a better rate of photosynthesis. If we look at these C4 plants have an overall leaf structure that is different from a typical C3 plant. And these plants, they are going to have cells near the leaf surface, which is where the oxygen as well as the carbon dioxide would be entering the leaf. So keep in mind that our atmosphere contains both types of molecules, which would then enter through the leaves' pores. Now, our Rubisco enzyme is not going to be the first molecule to actually grab hold of that carbon dioxide. Instead, we're going to have a different enzyme. This enzyme's name is Pep Carboxylase. And Pep Carboxylase is going to fix the carbon. So it will be the first enzyme that grabs that carbon dioxide. It's going to hook it to a 3-carbon molecule, and we're going to end up with a 4-carbon molecule, which is a stable molecule called oxaloacetate. This Pep Carboxylase is an enzyme that's not going to be affected by oxygen gas. This enzyme is going to bind the carbon dioxide in the presence of the oxygen gas, and at this point we do not have to worry about increasing the carbon dioxide concentration. That 4-carbon molecule is going to be rearranged a bit and transported further down in the leaf, and this means away from where all of that oxygen is going to be in the atmosphere. Once we get it transported into a different cell, more buried in the leaf, we're going to cut the carbon dioxide back off of the 3 carbons, and this is where the Rubisco enzyme is going to be present. What we're doing is we're essentially releasing the carbon dioxide right in front of the Rubisco, and there's not going to be any oxygen around. This means that the Rubisco enzyme is going to certainly grab hold of that carbon dioxide and then run the Calvin cycle and produce the sugar or the G3P that we want. So notice what we've done is we've collected the carbon dioxide at the surface of the leaf with an enzyme that's not affected by oxygen gas. Then we will transport the carbon dioxide down further in the leaf and re-release it right in front of the Rubisco enzyme. When we do this, we are increasing the concentration of the carbon dioxide and also decreasing the concentration of the oxygen gas where the Rubisco is located at. Here's just another look at it. We can see that where the carbon dioxide is coming in is going to be different in location from where the Rubisco is going to be acting. So the Rubisco would be acting down here in the Calvin cycle, which will be much further down in the leaf structure. If we have plants undergoing this C4 metabolism, they will have a higher rate of photosynthesis. A higher rate of photosynthesis because they're going to have a much lower rate of photorespiration. If you do any gardening outside, whether it's vegetable gardening or just flower beds and things out front, there are some plants that appear to grow much faster than other things. So weeds are one of those. Weeds seem to grow quite fast compared to the little plants that you're trying to grow. Grass is similar to that. Those plants not only appear to grow faster, they do grow faster. And that's because weeds and grasses are typical C4 plants. The flowers and the vegetables that we try to grow, those are our C3 plants. So they are struggling much more in their photosynthesis because they do have that process of photorespiration working against them. Now, one other alternative way that carbon is fixed is with plants that are in the desert. So these plants have what we call crassulation acid metabolism, or we refer to them as CAM plants. The problem that desert plants have is that water is not available at high concentrations. Not only that, but during the day it's going to be very dry, so we even have a very low humidity. If plants are going to fix carbon, they've got to be capable of collecting carbon dioxide. And the only way to collect carbon dioxide is to open the pores on the leaf surface. If those pores are open during the day in the desert, though, we run the risk of losing a lot of water out of the pores. So we would really like to keep those pores closed to maintain any water that is inside the plant. What these plants are going to do is that at night, when temperatures in the desert are quite cool, we will open the pores of the leaf and the carbon dioxide will come in. When that carbon dioxide enters the leaf, we're going to use that Pep Carboxylase enzyme again, and it will do the initial fixing of the carbon. It will convert that carbon into a C4 form. Okay, so very similar to what we were doing with our C4 plants. Then that is just going to sit that way as long as it's nighttime. When the sun comes out, the pores will close so that we do not have to worry about water loss. At this point, the light reactions are taking place, and then we're going to re-release the carbon dioxide, and the Calvin cycle will then be able to run. So the Calvin cycle will take the products from the light reactions, the NADPH and the ATP, and it will use them to reduce that carbon dioxide. Notice in this case that our carbon was collected at night, but we don't actually use it for the Calvin cycle until the daytime. So we have a time separation or a temporal separation of when we're collecting the carbon dioxide, when we're actually using it. If we compare this to the C4 plants, we have what we typically call a spatial separation of carbon dioxide collection and utilization in the Calvin cycle. So we collected it in one location, we use it in another. In the case of the CAM plants, we collect it at night and we use it during the day, but all of that is taking place inside the same cell. Notice in both of these alternative photosynthesis approaches, we have an initial enzyme that is not Rubisco, it's Pep Carboxylase, that does the initial fixing of the carbon. That's not to say that Rubisco doesn't have a role in these plants, because it certainly does. It will just be later on in the process, because the Rubisco will be running the Calvin cycle when and where it does ultimately take place.